Hello and welcome everyone to another episode of Biology with Arseni. It's been 2 months since I have made an episode, and that was for two reasons. The reason number one, we had some technical problem. We had overcome these technical problem. We had a better gear, a better set, and all new gear. So that's a great thing. But after this, there was a big problem. I broke my hand for around a month and a half, and after a month and a half and 16 stitches later, I'm back. Biology with Arseni back. We'll continue what we are doing. We will continue what we are going to say everything concerned to the biology. We will continuing. Um, as always, this is for everyone who's been supporting me for the last 2 months. Thank you so much. For the family, for the friends, thank you so much. Thank you for your support. My friends at work, thank you for support. My friends at everywhere, thank you for the support. I'd like to extend all the appreciation for the people abroad who asked about me, about my health. Thank you so much. Now, we are back. We are here to continue what we are doing. Everything concerned to the biology. Mainly, the thing is, in the past few days and the past 2 months, the idea was that people were having trouble concerning the type of testing that we are having in Egypt, whether they are within the national department or they are in the American diploma, especially the American diploma, especially after canceling the SAT exam and the ACT exam. Now, the grade 12 students, they are stuck with the EST. It's a better chance for grade 12, grade 10 and 11, but for grade 12, well, you are stuck with the EST. Now, the thing is, the past videos and all of the next episode is going to be targeted the EST exam and the EST biology exam on particular. It's going to help you to get a better scoring and a better insight on how to have another insight on how to be familiar with the biology found in the EST exam. I'm gonna try to put some kind of exercises concerning the exam itself and how you answer these questions. So the people who are taking EST biology here in Egypt, pay attention please. The student of the national department who are having their exam, well, no problem, pay attention please, because we are going to have other episodes that it's going to help you down the road. um, for the people who are waiting for the Arabic episode, there will be another episode for that topic in Arabic. If you are don't like the English, or you are not familiar with the English, well, you're going to have another episode as well, and it's going to be great. So, without any further ado, we will start after um, after the intro. Thank you. So today's session is about nucleic acids, and since we started our episode, we have talked about a full type of major things. This is number four of the major bioorganic molecules. We have talked about carbohydrates, we've talked about lipids, we've talked about proteins, we've talked about enzymes, how the enzymes work, and we reach our final and last biomolecule that we have within the living organism, which is the nucleic acid. Now, nucleic acid is a big bunch of things, but we are going to simplify it as much as we can because we are going to have episode later to talk about details within the nucleic acid. And as we always know, these nucleic acids, since beginning, they are a kind of polymers, and that polymer itself is made from a kind of monomer that is called a nucleotide. Now, nucleotide, they are a type of monomer that has other several pieces from the inside, and they have a general structure. Now, these nucleotides, they have a general structure of a sugar, which made of 5 carbon sugar that we call a pentose sugar. Remember, the word pent, it's meaning for 5. Okay, so the word pentose, the syllabus O's at the beginning, the OSE at the, I'm sorry, at the end, it is for the sugar, and it has a nitrogenous base and a phosphate group. Now, we can find it here in that picture. This is a general structure of a nucleotide, okay? A phosphate, a pentose sugar, and a nitrogenous base. Nucleic acid, they have two types, okay? And these two types, you are either talking about DNA or you are talking about RNA. Well, both of them, they are nucleic acid, but there are huge differences between them because, of course, of the name DNA and RNA, we're gonna know what is the naming process. For the DNA, it called the deoxyribonucleic acid. Now, it has a deoxyribose sugar. Now, the word deoxy means it is missing an oxygen from from the sugar itself. That's why we call it deoxyribose. Okay, on the RNA, it is called the ribonucleic acid, and it has a normal ribose sugar, no missing oxygen or whatsoever. So it has the ribose word itself. And after that, there are the bases itself. Now there are 4 types of nitrogenous bases. Within the DNA, we have the adenine, the guanine, the cytosine, and the thymine. On the RNA, there is also 4 types of these nitrogenous bases. We have the adenine, we have the guanine, we have the cytosine, but there is a slightly big difference. We have a uracil over here instead of the thymine within the DNA. Now, the other difference is the DNA is a double-stranded, and there is a missing D over here. Let's put it. Okay, and the RNA is a single-stranded. As you can see over here, this is the main difference. Okay, now if you took a look at the deoxyribose over here and the ribose over here, you find this is a hydroxide group over here, and here there is a missing oxygen here. That's why we call it deoxyribose. This is the missing oxygen over here, okay, that make it deoxyribose. You can see this is a double-stranded DNA, okay, and they have a base pairing. They are complementary to each other, okay, they are connecting to each other, and we're gonna know why they are connecting to each other. Now, on the other hand, the RNA, it's a single strand. There is no kind of bonding or whatsoever, just a normal strand, and that's it. Now, the connection between the basis itself, okay, between the nitrogenous basis itself, it's what you see here in between. This is the nitrogenous bases, they are connected to each other, okay, and we call it a base pair. Okay, why a base pair? Because I have 2 bases, that's a pair, they are connected to each other, so we call it a base pair over here. So moving now, we have the 5 nitrogenous bases over here. Now, these 5 nitrogenous bases over here, we have the adenine, we have the guanine, and we call them the purine or the heavy bases. Now, the purine, they have a double rings, okay? Whether you are talking about adenine or they're talking about guanine, they have a double rings over here, and that's why we call it the heavy bases. They have a huge molecular weight. Okay, and the brimidines, which are the uracil, the cytosine, and the thymine, okay, we call this the brimidines, okay, and they have 1 ring, okay, or we call them the light bases. Now, within the DNA, we have these base pairing. They are connected to each other by a hydrogen bond, okay, but how these strand, or how the strand is connecting one base after the other, it is so simple. Now, in order to understand how one base after the other connecting to each other, now we have to understand something. Now, if we are having here this is the pentose sugar, okay, this is a phosphate group over here, and that is a base. Okay, this is the base. Okay, and that is the sugar. Okay, so the next in line, it connects the sugar to a phosphate group. Okay, and the sugar continue. Okay, so it continues over here like this, and this is the base. So it's a kind of a bond between the phosphate and the sugar. Okay, so this is how they connect to each other. It's another example of how covalent bond can help in connecting things with each other, and how they help to stabilize our DNA. Now, this hydrogen bond also helps in having the DNA tolerant to somehow high temperature that up reach to a 100 degree. At 100 degrees Celsius, the DNA base pairings start to break down, and you have two separate strands. Okay, so these pairing, they work with the adenine is bonding with the thymine or the uracil, and the guanine bonds with the cytosine only. Other to mention that the number of the adenines equal to the number of thymine. You don't have an excessive number of adenine and a shortage in number to the to the thymine. No, that doesn't happen. You have, let's say, 10 bases of adenine. On the other hand, you are going to have a 10 bases of thymine. Okay, the number of the guanine equal to the number of the cytosine. This is also the same as the adenine and the thymine. You cannot have an extra number of one base an extra number of bases to another number of bases. No, that doesn't work this way. You will have the same number. Okay, so these how the bond with each other, and how these bases bond with each other. Now, another thing that is so important, now the DNA strand, they are anti-parallel from each other. Okay, like you're working a hero and anti-hero. Now we have a strand that goes from the 5 dash to the 3 dash line. On the other hand, the other strand, it goes from the 3 dash end to the 5 dash ends. Okay, this is how, if we return back to that diagram over here, this is how the base pairing work. You cannot have the same base pairing, or you don't have the same strand going to the same direction. Okay, they won't base pairing. That the DNA doesn't work that way. Okay, so in order to understand how the DNA work, well, it took a lot of time. Actually, the DNA had been discovered a long time ago, but the actual image that came for the DNA came to the 60s, okay, by a scientist who her name was Rosalind Franklin. Okay, now Rosalind Franklin had give the first X-ray image for the DNA. After that, in after that, Watson and Crick, okay, they both developed the double helix for a double helix shape for the DNA. Well, after that moment, humanity didn't look back, because DNA was a key for lots of things, a key for lots of study. Till today, we use it in too many things. Okay, we use it for, let's say, identifying people, we use it for solving crimes, we use it for understanding how the human cell work, and how a bacteria work, how viruses work, lots of things that the DNA help us to understand in order to go and have an insight within what the other organism, the living organisms, whether they are human or not human, work. So that is going to be all for today. Thank you all for watching. Thank you again for the people who have helped us to overcome this 2 month period, and I promise you from now on we are going to have our episode an episode for every week. So with us for the next episode from Biology with Arseni. The next episode we are going to talk about cellular biology. So stay tuned for the next episode. Thank you so much. See you later, everyone, and goodbye.